I'm Da-Neng Wang from New York University School of Medicine, and then some of my colleagues, David Sauer and Jim Mason, are here on the teleconference with us. So today I will tell you about our ongoing work related to the SLC13A5 gene and the NaCT transporter protein, how we efforts we make in the lab to understand the mutations that cause the disease, and also effort in developing a small molecule screen assay. So this is a table Kim circulated with the mutations that cause the disease. And then the most common one is the glycine 219 to arginine mutation. And then we are making efforts in the lab to understand how each of these mutations abolishes the NaCT transport activity and then causes the disease. So all this started from a paper published by a pager 3 years ago, where they observed the distribution of the expressed protein in the cell and on the cell surface. So they found some of the mutated proteins, the mutants, would express in the cell, and then but they would not go to the cell surface, to the plasma membrane. And the others were expressed and did go to the plasma membrane. And they called the first one type 1, that is trapped in the ER, and then they thought this was due to either the protein stability or protein integrity. And then second type, they called type 2, they are located correctly in the cell surface, on the cell surface, in the plasma membrane, and then the defect was due to transport activity. And then we wanted to analyze this further. So the place we started was to understand from the point where how the transporter protein transports citrate. And then it's believed that protein works in such a way. So there are two conformations. One is called an outward facing conformation. One is called an inward facing conformation, and then citrate binds from the outside. And then this domain is called the transport domain, moves down in the membrane, and then citrate is released. And then in our lab, a few years ago, we determined the structure of a NaCT homologue, bacterial homologue called VCND by crystallography. And then this is the crystal structure we solved. And then you see this is a scaffold domain or dimerization domain. This is transport domain that can move up and down. And then we followed Anna Pager's footsteps. So we try to analyze and classify all the mutations at that time. And then we further classified, it's not published, but we further classified class 1 into class 1, type 1 into type 1, type B. So type 1 would, mutations include those of early termination of phase or frame shift. And then type 1B are mutations that affect folding or dimerization of the protein. And then type 2A are mutations that near the substrate binding site or the sodium binding sites. Type 2B are those mutations that affect the movement of the transport domain. So that is, say, if the mutation is at this interface, it blocks the transition of this domain from this state to that state. So, so we could classify those mutations. And then for type 1A and type 2A type of mutations, I think the only therapy or only intervention people can have are probably gene therapy. However, for type 1B and type 2B type of mutations, small molecule drugs is a possibility that can probably help with protein folding or helps with conformational changes, like in the case of CFTR channel protein. So to that end, we started to search for a way to screen for, to develop an assay to screen for small molecules. So to have something efficient, we want something simple and can be monitored easily and cheap. So David Sauer in the lab had the idea of using E. coli. So this is based on a classical experiment done by Richard Lanski at Michigan State, where he realized the E. coli cells, bacterial E. coli cell, has only one citrate transporter. It's called CT. And then CT is only expressed when oxygen is not present, is highly inhibited or suppressed when oxygen is present. So therefore, he started an experiment, already this is almost 25 years, just to grow the E. coli cells, see if this, to demonstrate how tightly this gene is controlled. So only after something like 10, 12 years of cell growth, daily cell re-inoculation, cell E. coli growth every day, only after something like 60,000 cycles, he found one mutation that occurred in the promoter region of CT that allowed C2 express, and then allowed the E. coli cells to grow on citrate as a sole carbon source. So David had this idea, so to do this, to express NaCT in E. coli cells in order to screen for small molecule drugs. So we would, when there's no functional transporter in E. coli, so cell will not grow. So the media will remain clear. So if we can introduce the human NaCT protein in E. coli, the cell will grow, and then you will see that the media becomes less transparent, would have a higher OD. So if we can put in a mutated NaCT mutant, which is either type 2B or type 1B, so you would have a dysfunctional protein. And then if we add in small molecules, small molecule compound libraries, and then if one small molecule binds to the transporter and then can rescue the transporter to allow the transporter to be functional, and in the plasma membrane, that would allow citrate to come into the cells, and then the cell would be able to grow. So that we would have a very simple assay for screening small molecule drugs for this particular type of mutant. And then we can validate this in mammalian cells. So the key of the experiment is to be able to express the human protein in E. coli cells. So David first did a proof of concept experiment using an bacterial citrate transporter called TESS. And then when he put this in, without, this is empty vector, without this bacterial citrate transporter gene, cells didn't grow. And then when this TESS was introduced, cell could grow. So in principle, we could use E. coli to monitor a functional citrate transporter. And then we started to put in human NaCT gene into E. coli cells. And then we used a codon optimized gene in order to get higher expression level. And then we also used two fusion partners, which are small compact proteins, to be fused either at the N terminus or the C terminus of the protein. And then we call them the alpha and beta. And then after we introduced such constructs, everything grows in the same way. So without NaCT gene introduced, the cells would behave in the same way. So then we analyzed this, we found out in all the cases, we could now see NaCT expression. The NaCT, if it's expressed, should be somewhere here or somewhere here with fusion partners. So in all this constructs we tried, we couldn't get NaCT expressed. So then we went back to check this. Perhaps the major problem was we used a codon optimized human NaCT gene to get high expression. And then too high expression level probably was toxic to the cells. Therefore, it didn't express at all. So now we are doing experiments to express a wild type NaCT gene. When we have results, we'll report to you. And then the second half of the talk, I want to cover something on the mutations again. So this is a figure I showed you before, and then we could explain about 15, 20 mutations. But then there were some we didn't understand at all. For example, this one, D524 methionine mutation. This is far away from the substrate binding site, far away from the interface. We didn't know how this one would abolish substrate transport. And the reason we thought was probably because we only have one the structure of one state, not the outward, only the inward facing state of the protein, not an outward facing state of the protein. So we couldn't use modeling to understand this. This is to show this again. So this is a transport cycle. We only have a structure of this cycle, and then we don't have structure of that cycle. So mutations that blocks can be explained in some of the mutations can be explained in this state. We don't have that state. So for this, we in the lab we switched to another bacterial homolog. It's called LA-IND. So we did structure determination for this protein using a new technique called cryo-electron microscopy, which allows us to use purified protein to substructure directly without doing crystallization. And then this is the structure, one of the structures we got for this LA-IND protein. And you can see the dimerization domain, it sits in the middle, and then you have a transport domain moving in a different state. And then if you compare this new LA-IND structure in an outward facing state to the base-IND structure in the inward facing state, you see the transport domain move from a down state to up state, and then the substrate binding side faces this side. So indeed, we have this structure. In an outward facing state. And then we did modeling again. So we modeled the human NaCT protein based on a bacteria, the bacterial protein structure. The sequence identity is about 25 to 30%. And then now we have a model for both states of the human protein, and we can analyze the mutations in a much more thorough way. For example, this D524 M mutation in the inward-facing state is there. In the outward-facing state, it goes there. So that mutation allows a hydrophobic patch to be formed at that interface, and that probably blocks the transport. And then from the list Kim sent us, we reanalyzed everything, and then we can classify them in a much more confident way. However, there are still 2 or 3 mutations, including the most common mutation, glycine 219 arginine mutation. We cannot quite say for sure why they abolishes transport. This one is kind of close to the substrate binding side, but it's not that close, so we do not quite know. So that probably requires us to solve a structure of this protein, this protein mutant, to see exactly how this such a mutation harms the protein and abolishes transport activity. So this is a acknowledgement slide. So this is our lab people, David Sauer, is postdoc who is in the audience, who is the leading author of the of the work with help from Jimei Song and Jennifer Martin. And then we needed a lot of cryo-EM time for this data collection, and we used 3 cryo-EM facilities, and then these are the funding sources. Thank you.
Thank you. That was so wonderful.